Within the chromosome, DNA is packaged into chromatin. Chromatin consists of DNA, structural histone proteins, and non-histone proteins. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of 146 base pairs of 2 superhelical turns of DNA wrapped around a core of 8 histones. The histones are responsible for maintaining the chromatin's shape and structure.
Double-stranded DNA is a polymer of 2 strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell.
One common error is base mismatches. Each nucleotide contains a base, and during DNA replication, the enzyme DNA polymerase is supposed to bring in the right partner to pair with every base on each template strand, adenine with thymine and guanine with cytosine. But about once every 100,000 additions, it makes a mistake. The enzyme catches most of these right away and cuts off a few nucleotides and replaces them with the correct ones. And just in case it missed a few, the DNA in just one of your cells gets damaged tens of thousands of times per day. The errors come in different forms. Sometimes nucleotides, DNA's building blocks, get damaged. Other times nucleotides get matched up incorrectly, causing mutations. And nicks in one or both strands can interfere with DNA replication, or even cause sections of DNA to get mixed up. But DNA can get damaged after replication too. Lots of different molecules can cause chemical changes to nucleotides. But others are molecules that are found in cells naturally, like hydrogen peroxide. Certain chemical changes are so common that they have specific enzymes assigned to reverse the damage. But the cell also has more general repair pathways. If just one base is damaged, it can usually be fixed by a process called base excision repair. One enzyme snips out the damaged base, and other enzymes come in to trim around the site and replace the nucleotides. UV light can cause damage that's a little harder to fix. Sometimes it causes two adjacent nucleotides to stick together, distorting the DNA's double helix shape. Damage like this requires a more complex process called nucleotide excision repair. A team of proteins removes a long strand of 24 or so nucleotides and replaces them with fresh ones.